Welcome my students. I discuss about gene mutation. There are some difference between gene mutation and chromosomal aberration or chromosomal variation. You know gene mutation in genetic level, but in chromosomal aberration or in chromosomal variation, which is structural or numerical change in chromosome level. Our discussion about gene mutation and DNA repair. Let us discuss about what is gene. You have some idea about gene, that a gene is a basic physical, functional, structural unit of heredity. Okay, but there are modern definition for gene, that a gene is a sequence of nucleotide of DNA or RNA which synthesis gene product, either RNA or protein. That is gene. So you have some idea about gene and about genetic material. Our discussion, gene mutation. Mutations means anything change in gene, anything alternation in gene, which is transcribed, which is transferred from one progeny to another, to progeny to offspring. So our discussion about mutation is inherited change in the genetic material, provided genetic variation that allows organism to evolve. There are many types of mutations, that is somatic mutation, germinal mutation, spontaneous mutation, induced mutation. So our first term is somatic mutation. What is this? Somatic mutation means when the mutation or alternation present in somatic cell. Here the mutant in phenotype only descendant of that cell, that is somatic mutation in somatic cell. But in germinal mutation, when the mutations occur in reproductive cells or gametes, here the mutations, effect of this mutation transfer from one generation to another generation also. There are one mutation is called spontaneous mutation, when the mutations occur without any mutagen or without any physical or chemical agents, that is spontaneous mutation. Here many spontaneous mutations occurs. In bacterial cells there are every 10 to the power 8 or 10 to the power 10 cells happen spontaneous mutations. And in eukaryotes, that is 10 to the power 8 or 10 to the power 6 number of cells. One of these 10 to the power 6 and 10 to the power 8 number cells, there is one spontaneous mutation. And our next discussion is induced mutations, when the exposure of an organism such as physical and chemical agents cause mutation, cause changes in DNA and RNA, is called induced mutation. Many physical agents, that is UV ray, eradication, intercalating agents, that cause the mutation called induced mutation. And other type of mutations are forward and reverse mutations. Forward mutations means the mutation of wild type of gene. One wild type of gene here change into mutated genes, that is forward mutations. The mutations of wild type of gene to form a result of mutant phenotype, that is genotype of wild type of gene, and that is mutant phenotype. That is wild type and that is mutant type. This is the phenotype of wild type, and this is the genotype of wild type, and in this region, that M region is mutated. So this is forward mutations. One another mutations occur, that is reverse mutation. When this mutated phenotype back into wild type phenotype, that is called reverse mutations. Reverse mutations are two types. One is back mutation, one is suppressor mutations. Two phenomena, two incidents occur when the first one, this mutant phenotype become the wild type. This is the back mutations. Okay. In the second type, there are another mutation in another region. This is S here. This suppresses the effect of M, and this phenotype like genotype. This phenotype like wild type genotype. That is called suppress mutation. When mutations occur in single pair of nucleotide is called point mutations. Point mutations are many types. I discuss about this. That is, point mutation also cause severe damage in DNA. That is transition mutation. When single base pair of this region, this base pair, that AT become GC, A become G, so purine become purine and pyrimidine become pyrimidine. This is transition. When purine change into purine or pyrimidine change into pyrimidine, this called, when purine to purine and purine to pyrimidine change occur, this is called transition mutation. Another type of mutation is transversion mutation. You saw in this picture that C into G and G into C, that pyrimidine change into purine and purine change into pyrimidine. This is called transversion. And another type of mutations, another type of point mutation is missense mutation. You look in this figure. Here mutations occurred in this region, AT region. It is become GC. So transition occur. What is the effect of this transition? That it produces serine, glycine, lysine, phenylalanine, and tyrosine, and but here one amino acid change, it is produced glutamate. Missense mutation change one amino acid to another. So here the codon also change. Here the codon is AAA and here the codon is GAA. Okay, so that is missense mutations. Next type is nonsense mutation, where the mutations occurs and the product of this mutation, the result of this mutation, it produce stop codon and the transcription and translation stop. So the translation stop. Here you look at that AT it transfers into TA. For this reason, the glycine here the AAA, the glycine UAA transfers into stop codon. Here number one change give result for stop codon. So the translation stop. This is this is the nonsense mutation where the translation stop. Another three types of mutations, point mutations, are neutral mutation, silent mutation, and friendship mutation. These are also very easy to understand. That in neutral mutations there are no change in amino acid sequence or no change in codon, but there is no change in effect of protein synthesis, where in this this AT transferred into GC, but it produces lysine. Here it produces arginine. Lysine is positive charge amino acid, arginine also positive charge amino acid. So the total effect, the total protein synthesis criteria, the protein functioning is same. So this is neutral mutation, change from one amino acid to another amino acid with similar chemical properties. So there is change in codon from lysine to arginine, but the function of the protein is not changed. Another is silent mutation. That mutation occurred, codon altered, but the result of the amino acid sequence in translation do not alter. Here had the AT change into GC, but AAA codes lysine and AAG also code lysine. So there is no change in amino acid sequence. It is silent mutation. And another important mutation is friendship mutation. What is this? Where one deletion or one insertion in this DNA sequence cause the shift of the frame of the codon, okay, and change in the amino acid sequence in transcription, in translation. Here the GC inserted here. So here the protein sequence is serine, glycine, lysine, phenylalanine, threonine, but after insertion of GCs, the codon change. 3, 3, 3, 3, the codon sequence change and produce serine, glycine, glutamate, isoleucine, and tyrosine. That is friendship. Total frame is shifted from one amino acid sequence to another amino acid sequence. Okay. One question arises: why these mutations occur? Due to the external or internal or chemical effect. But what is the change? What is the change internally, molecularly? Change occur in many regions, many bases. These changes is called tautomeric shift. Hydrogen atoms can move from one position of purine or pyrimidine to another position. For example, for amino group ring nitrogen, such as chemical fluctuation, this chemical fluctuation is called tautomeric shift. Generally, 3 and 4 position of the pyrimidine and 1 and 6 position of the purine alter into enol form to keto form or keto form to enol form. Here, that is the keto form thymine. This is keto form thymine. It changes into rare enol form. Okay, that is amino form cytosine. It changes into enol form. It changes into imino form. So this change into imino form. Okay, so that is the tautomeric shift. For this reason, mutations occur, and internally that change into various forms. In the third example, that amino group change into imino group, and here the guanine change into the guanine, the keto form of guanine change into enol form. Okay, change into enol form. Okay, the keto form of the guanine change into enol form. What is then? What is there? What is then the cytosine? Cytosine after tautomeric effect. The cytosine and guanine are their rare immuno enol tautomeric forms binds into cytosines binds into adenine. Cytosines binds to adenine, not cytosine to thymine, and also thymine binds to guanine, not to adenine. This phenomenon occur, this incident occur, so the mutations occur for this tautomerism. Here in this picture you can detect how it occurs in this region. After replication in this region, total the change is occur. What is mutagen? Mutagen are physical, chemical agents that causes mutations. Many X-ray, gamma ray, alpha particles, cobalt 60, cesium 136, UV can cause the damage in DNA. This is the physical mutation. Physical mutagens, and the chemical mutagens are base analogs, intercalating agents. Here the base analogs. What is the base analog? The mutagenic base analog have the structural similarities. Structural similarities with the normal base. Structural similarities with the normal base. Here 5-bromouracil, this is keto form, like uracil which binds with adenine. Here 5-bromouracil also binds with guanine. Okay, this is the base analog. Here some chemical mutagens which calls mutation, that is alkylating agent, base analog, alkylating agent, okay, intercalating agent, deaminating agent like nitrous oxide, and hydroxylating agents like hydroxylamine. Second one is deaminating agent. What is it? Some mutagens have potential to remove the amine group. Such mutagens is called deaminating agent. Nitrous oxide causes the oxidative deamination of the amino group in adenine, guanine, cytosine, all of these. Deaminating. Here, this is the amine group and it is become keto group here. Cytosine become uracils here. So that is the change in basic level, in base level, and guanine becomes xanthine. After the incorporation of nitrous acid, oxidative deamination occurs and guanine change into xanthine and it binds to cytosine. So the mutation occurs, and this is the nitrous oxide, the deaminating agents, chemical agents also causes mutations. Some intercalating agent which binds to in the group, major group or minor group, instead of which binds in the major group and minor group, and this is causes the DNA mutation, such as proflavin, acridine, ethidium bromide, between the adjacent base, it is the one base, it is bad. It is intercalating agent. Between the adjacent base, it binds and causes mutation. That is intercalating agent. Generally, intercalating agents are used in laboratory, in electrophoresis experiment, etc. some alkylating agents. Some alkylating agents are chemicals that donate alkyl group to the molecules, and it causes mutation. That is nitrogen mustard, methyl and ethyl methane sulfonate, chemicals that have multiple effects on DNA. Alkylating agents include types of mutation, include transition, transversion, frame shift, and even chromosome aberration also occurs for this effect, that is alkylating agents. Okay. How to detect mutations? How to detect mutations? Muller experiment with Drosophila. You know that he got Nobel Prize for this experiment. He used two types of chromosomes. One chromosome is CLB chromosomes. Muller constructed it CLB chromosome. It had three regions. Muller's experiments with these chromosomes and the effect of X-rays become possible, developed a simple, accurate technique that could be used to identify lethal mutations on X chromosome of Drosophila. This technique is called CLB method. Is performed with female heterozygous and a normal X chromosome and altered X chromosome, the CLB chromosome that Muller constructed specifically to use his experiment. What is CLB chromosome? Why the name CLB? C for crossover suppressor, refers to a long inversion that suppresses recombination between CLB chromosome and structurally normal X chromosome. L refers to recessive lethal mutation, in CLB chromosome, and B refers to the bird eye phenotype. You know that the bird eye phenotype, and in bird eye phenotype there are a duplication. In bird eye phenotype there is a duplication of 16A region. There are a duplication in 16A region. Okay. Now this bird eye phenotype for the B. So CLB chromosome have three regions, have three specific genes, C, L and B. Here the female contain one CLB chromosome and one normal X chromosome. Okay, it cross with irradiated X-ray incorporated were eradicated males. That X chromosome, this male with eradicated effect on this chromosome have X-ray was inoculated in this region. Okay, so there are some. So the experiment, female heterozygous with CLB chromosome and mated with eradicated males. Eradicated means the effect one X-ray for this, that the for the detection of mutation expose X chromosome, expose male chromosome into X-ray. So this eradicated X male, here the heterozygous female contain one CLB chromosome and one normal X chromosome. Okay, cross with eradicated X chromosome and normal Y chromosome. Four phenotypes. This is the 1, 2, 3, 4 phenotypes are liberated or produced. In first there is X from here, that CLBX and X-ray eradication. Okay, and there is normal X and X irradiated X, and number 3 is CLB and dies. This is X-linked recessive character, so it dies. Okay, this is normal X and Y chromosome, that is alive. 2:1, 2 female 1 male, there is 2:1 ratio. Then in the second progeny, CLB female progeny, this genotype was met with wild type. Okay, met with wild type male. So what is the progeny? That is both wild type X and CLBX. Here mutated, that is mutated due to the X-ray, that is here mutated X chromosome, and wild type X chromosome, this is also female. But one phenomenon, that is CLB and normal Y, X recessive, so this genotype dies because this CLB is recessive character. But detect mutation for this genotype, that here the mutated X chromosome with normal Y chromosome dies if lethal mutation was introduced. So the mutation caused the lethality, the mutation caused it incorporated the lethal genes in her, and this genotype dies. Okay, so this is the CLB method. So our last topic is DNA repair. After mutation, many DNA disintegrate, many DNA incorporated faults base. So what is the repair mechanism? It is the very simple. First repair mechanism is light dependent repair or photoreactivation. The enzyme called photolase, which this is the thymine dimer, here 2 thymine is occurred, this is the UV radiation, this is the thymine dimer, and photolase binds on it, and lase binds on it, and incorporated optimum or right base pair on the DNA base pair, and it's repair it. In the second types of DNA repair is the excision repair. Here DNA repair endonuclease containing enzyme complex binds to the excision site of the damaged DNA. In the second step, the polymerase fills the gap, and third, the ligase completely repair it. Okay. So in this there is a false base pair, deamination of cytosine into uracil. So deamination occurred. So first step here, glycosylase enzyme binds to the DNA strands, and it excises the uracil, and after that sugar phosphate removed by AP endonuclease and phosphodiesterase. The single strand break and DNA ligase seal it. So this is the excision repair, and that's all for this class. Thank you.